Hello and welcome to GeneLab Chat. Today we'll be interviewing Dr. Yang from the University of Saskatchewan. Dr. Yang and colleagues use publicly available astronaut data to assess gene expression profiles that could be used to monitor the health of space travelers. In this interview, Dr. Yang describes his research and how the GeneLab data systems and analysis working groups contributed to his reanalysis of Open Science Dataset 174. Thanks for joining us, Dr. Yang. I just have two questions today, starting with, what were the most important findings of your study?
First, I would like to thank you all, for giving this opportunity. It's a big pleasure to have this opportunity to talk a little bit of our research. In 2010, we initiated a research program on gene expression and correlation, which is based on the observation that any physiological or pathophysiological process requires the coordination of multiple genes. We propose to use genes that are differentially expressed and significantly altered in correlation to monitor health conditions such as cancer. But as a fans of NASA space program, my colleagues and I also decided to explore whether we could correlate genomic variation with the health conditions for astronauts. With the public data set from GeneLab and AWG, we conducted a whole genome analysis on gene expression and correlation and identified 32 genes that could be used to monitor astronauts' health conditions in space. Thank you.
Thanks for that. That's very important research, I believe.
Yeah.
And my second question, how did the GeneLab data systems or the AWGs help you assist in your work so you could achieve these goals?
I like to say that we could not complete this research without the strong support from GeneLab and AWG. We contacted the GeneLab several times during the research and to make sure that we did everything correctly and were on track. We also communicated with GeneLab and AWG on our results and sought their experts' opinions. I strongly hope that GeneLab and AWG can release more data sets and continue supporting our research to validate this monitoring approach. As a graduate from a lab that once was involved in NASA's space protein crystallization program, I wish I could also make my tiny contribution to NASA's space biology program. Thanks.
Thank you, Dr. Yang, and I assure you there will be more data sets coming. We appreciate the time you spent to share this information with us and in your research, also with our viewers. And to our audience out there, you can learn more about this publication, how to join the analysis working groups, and access the data set analyzed in this work with the links provided in the description section below. Thank you.
Thank you.